Welcome to my channel. I'm Charlie. So today video is a little bit different. Just a record of my final year project before I forget everything. So bear in mind I'm not top student but not bottom. This is a 2-1 standard project. Anyway, let's start. My final year project title is Assessment of Protocol to Generate Perfusable Vascular System from Endothelial Cells in Vitro, meaning I will have to find out a better methodology to build a vascular system, to build a perfusable blood vessel from this material, which is endothelial cells. In vitro meaning outside of human. So let's start to the main topic. The aim of the project is to investigate better methodology to build perfusable engineered blood vessel for the usage in VML. What is VML? VML is volumetric muscle loss. It's a substantial loss of skeletal muscle and impairment of the regenerative capability, which means the lost skeletal muscle will not grow back to its original size, and the whole human body have muscle, I would say. It can cause by trauma, surgery, or degenerative diseases. So why it is important to have 3D cell culture? Why we can't use just animal model or 2D cell culture? The main reason for this is 2D cell culture and animal model do not fully reflect human physiology. They do provide some understanding of human physiology, but ultimately, they do not fully reflect human physiology. Inappropriate model can give inaccurate positive result and failure in the clinical trial or unexpected side effect in human. This brings us to the urgent need of creating physiologically relevant 3D organoid as cells in the human body perform bioactivities in response to the 3D microenvironment. Often, cell necrosis happens in the center of the large 3D organoid. Necrosis means the die of the cell. Due to the lack of functionalized vasculature for oxygen, nutrient transport, and waste removal, functionalized vasculature become the key to fill in the research gap. Literature search on recent 4 years publications were done from Nature, Wiley Online Library, SES, Springle, PubMed, Frontier, MDPI. I don't know how to pronounce it. Sorry. Science Direct. Here is the summary of methodology to sort out useful papers. I figure out the importance of perfusable vascular system in organoid and focus on the search in creating vasculature. Next, I decide which cell to be the focus and group the printing methods. Then I list out the strength, weakness, and solution of different printing method to weigh which method is more appropriate. Lastly, I suggest a reliable method to build perfusable vascular system for the usage in VML. Human-induced pluripotent stem cell is a source of cells considered in this review due to its wide possibility. It can derive from a healthy or a sick adult somatic cells, and it can differentiate into different cell types. Tissue engineering by self-assembly, decellularized vascular graft, 4D bioprinting are among the approaches used to create engineered blood vessels, but our preference here is to build vasculature for the use in the VML. And due to the time limitation, only 3D bioprinting and electrospinning are well explained in this presentation. Tissue engineering is a multidisciplinary field where it integrates advances of bioengineering, biology, material science. The main cells in building vasculature are endothelial cells and parasites. Bioink is a fundamental liquid that composed of living cells and material to build scaffold for the interaction of cells. It can have a wide variety of formulation, depends on composition of each material. It can be natural polymers such as collagen, alginate, or synthetic polymers such as PEG, PCl. Now we will talk about 3D bioprinting. Imaging techniques such as computerized tomography, which we call CT scan, provide a hierarchical architecture of blood vessel. And this CT scan image was then translated into CAD image, which is computer-aided design CAD drawing, so that it can read by a 3D bioprinter. Layer by layer 3D bioprinting is suitable to use in vasculature printing because it affords multi-layer construct with thickness of 50 micrometer to 1000 micrometer. Multi-layer coaxial nozzle, which is a main player in this technique. Fixation of the bioink usually done through cross-linking with ultraviolet light, enzyme, chemical, or heat to form blood vessel-like shape. For the strength of this technique are high mechanical strength, afford multi-layer construct, short printing time, 3D space affords cellular viability in vitro. The weakness of this approach are toxicity from solvent for the degradation, high shear stress from the nozzle, damage the living cells. Now we will look at electrospinning technique. This technique involves the use of high voltage power supply, syringe with metallic needle for polymer solution storage, and a metallic collector to collect the nanofiber. High electrostatic force applied to the polymer solution, and then the liquid would then cross-link with cross-linkers such as genipin to form solid nanofiber scaffold. Electrospun nanofiber mimic the architecture of extracellular matrix, which is ECM. Experimental results from recent publications shown the advantage of aligned nanofiber scaffold in attracting the endothelial cells. Study led by News Group revealed the relationship between nanofiber orientation, RACAAA, and endothelial cells proliferation. From the figure, we can clearly see that more fluorescent endothelial cells were attracted to the CA and AA. CA means circumferentially aligned aligned, and AA means actually aligned nanofiber scaffold compared to the randomly aligned RA scaffold. And you can see that endothelial cell in CA and AA show spindle-like shape being parallel to the orientation of the fiber, whereas the cells show polygonal shape on the randomly aligned scaffold. Another group led by Nakayama compared the degree of endothelialization in aligned nanoscaffold nanofiber or randomly oriented scaffold. This is immunostaining results. It showed that more endothelial cells were attracted to the aligned scaffolds compared to the randomly oriented scaffold. So the purple color show endothelial cell. I mean, the purple color show CD3. I forgot. Sorry. This study involved transplantation of the endothelialized engineered muscle to mice with VML. Confocal microscopy image, which is this, show higher isolactin expression, which show in this light blue color, which I mean higher expression in the aligned scaffold compared to the randomly oriented scaffold, such as higher perfusability in aligned nanofiber scaffold. The strength of electrospun aligned nanofiber are high mechanical strength of multi-layer construct, independent from toxic solvent for degradation. Nanoscale fiber mimic native size of ECM and capable to attract the native endothelial cells. The weakness of these electrospun aligned nanofiber scaffold are longer printing time, cytotoxic and genotoxic from the nanofiber's material. However, this can be solved by reformulation of the biomaterial through increasing the natural polymer composition, decrease the synthetic polymer composition. In conclusion, for the best of my knowledge, electrospun nanofiber scaffold is the most suitable method to be used in creating perfusable vascular system for the usage in VML. The main reason for choosing this approach instead of 3D bioprinting, as discussed in previous slide, is that electrospinning has less disadvantage as compared to the 3D bioprinting. Moreover, I suggest to provide the vasculature with growth factors such as VEGF, a help growing of the endothelial cells, heparin coating, as this can avoid coagulation, and bioreactor, which provide physiological relevant pulsatile pressure. And yeah, I mean, because I already forgot some of the knowledge, so it seems like I don't know much, but I mean, this is my final year project. I would like to thank my supervisor for his guidance throughout the project. Thank you.